Okay, it seems like it's working now. Eduardo's look good over there.
No, but I'm not sure where it's me. If somebody is better at checking that we're live, help me out. In my screen doesn't come up, but I'm not
We
sure.
are live.
We
Yeah.
are?
I've got a YouTube symbol.
Okay, great. Well,
Thank you,
thank you.
guys.
All right, so hello everyone, wherever you are. Good morning, good afternoon, evening. Welcome to Fine Seminar. My name is Lauren Hayes. I'm at the University of Tennessee at Chattanooga, and I'm hosting this week's seminar. Really excited to have. We have two speakers today, a collaborative seminar. So looking forward to that. Briefly, I want to highlight what's coming up, and I'm going to share my screen here. And so we got a really exciting group of talks coming up. Today, I'll introduce Hannah and Steve in just a moment. Next week, we have Loreto Correia, who is a professor in Chile, and she works on how masculinization morphotypes influence social systems and reproductive success, focusing on Daegu. So I hope people can make her talk next week. Today we have I'm really excited about today's talk because we have, as I said earlier, two people participating. We have Hannah Correia and Steve Dobson. And I just want to give a quick intro to them. Uh, Dr. Correia is a postdoc at Johns Hopkins University. Previously, she postdoced at the Harvard School of Public Health, and she did her PhD and master's both at Auburn University. And her master's was actually in statistics, and her PhD was in biology. Her research focuses on the interface between statistics and ecology. And I think we're going to learn a lot about that today. It's really interesting stuff. She focuses on things like causal methods to estimate sort of the consequences of environmental change and think about how this affects animal behavior. Um, Her research looks at, how behavioral changes influence not only ecology, but also planetary health. And we were talking about this a little bit earlier. Her research is focused on, using statistical methods to look at like how stigmas regarding disease like HIV play a role. She's modeled climate change and how that affects population dynamics in fish. And she's even delved into studying how climate change affects reindeer in Norway. So quite a diverse research program. Again, that interface between stats and ecology, really important stuff. She's been publishing well. She has over 20 publications. And what's exciting about this, she's publishing both in statistical journals and some of the really good biology journals like Proctoral Society B, Scientific Reports, and bio essays. She's going to be collaborating today with Dr. Steve Dobson. Steve is a professor emeritus at Auburn University, and he also holds or has held a fellowship at the Correia and Pluridisciplinary Institute at the University of Strasbourg in France. And I'm sorry I butchered the French. It's been a bit of a while for me on the French side. Um, Steve has had a prolific career. Research has been really diverse, focusing on things from behavioral, historical, and population ecology, population genetics. He studied a wide diversity of species, birds, mammals, and I think I even saw a mosquito paper in there, if I remember correctly. Um, so he's had over 150 publications in a wide variety of journals, has a book on why penguins communicate, the evolution of visual and vocal signals. That was published in Academic Press. And he's held positions in various countries, including, as I mentioned earlier, France, of course, the United States, but he's also done research in places far away as India, where he had a Fulbright fellowship. He's had positions not only in Strasbourg, but also Paris. Quite a prolific career. And as you'll see in the talk today, has started thinking about using, working with Hannah on mathematical modeling or statistical modeling to answer important questions in ecology. With that, I'm going to turn it over to the two speakers today, and I want to thank you both for being here. I'm looking forward to your talk.
Thanks, Lynn. That's a really nice introduction. Really appreciate it. Hannah and I are going to talk about multiple paternity, but we need to mention a few other folks that we're also collaborating with. One in particular is Ash Abibi. He's chair of the math and statistics department at Auburn University. And Ash has been a mentor to both Hannah and myself. Two other people that we've recently started working with are Lauren Hayes and Maddie Roberts at University of Tennessee, Chattanooga. So the work's going to include some input, especially from those folks. Slide, please. Okay. This just shows a diversity of different types of animals. And in species where organisms have more than one offspring at a time, we frequently see multiple paternities. So these are all species that within their populations, we see multiple paternity. The one on the upper left looks like a mouse, but in fact, it's a marsupial. The lizard's a common lizard from Europe, a tree swallow, and some sunfish. And all of these have high rates, fairly high rates of multiple paternity. Slide, please. So what do we know about multiple paternity? Usually what happens is we go out in the field and we sample individuals in nature and look at usually mothers, but often we don't know fathers. And we get, say, a mother and a certain number of individuals of her offspring in, basically a litter, a clutch, or a brood. So we know what the brood size is, and often we know what the mother is, not always. Take some tissue samples and take them into the lab. And so far, a lot of the work in the lab has come from analysis of microsatellites, hypervariable loci. And from that, we use statistical software to figure out who the father is. Sometimes we don't know the mother, and we can get some additional insights on that. And when we're done with the lab results, we can see how many fathers there are for a brood, and that's where we get an idea of which broods have a single father and how many have multiple fathers, and we can look at the proportion that have multiple fathers. Slide, please. So in looking at multiple paternity, the first question that we might ask is, how much multiple paternity should we expect? And the first principle that I'm going to look at is multiple paternity ought to deal with how many mates, usually females, have, and then how many offspring do they have. In order to get multiple paternity, of course, if there are more and more offspring, there's a higher and higher chance that more than one sire will be represented. And also, as there are more and more mates, we'd expect more multiple paternity. So that's pretty simple. What we want to do is come up with a null model so that we can test real data versus what we would expect under some sort of basic assumptions like random mating. So the little formula on the lower left here that I put is actually a combinatorial for how much multiple paternity we might expect given a certain number of mates and a certain litter, clutch, or brood size, assuming that there's an equal chance of paternity for all of a female's mates. Now in the field, Unfortunately, we often don't know M, the number of mates. But if we did, if you look here on the right, we can figure out how much multiple paternity we ought to expect under our assumption. So I've just plotted out here using the combinatorial. If you have 2, 3, 4, 5 mates for a female, how much multiple paternity you would expect over different brood sizes. Okay, from lab data, we don't know how many males actually mated with the female. We just know how many of them fathered offspring. But we can see from this that we actually get a curvilinear pattern of expectations. Okay, and in fact, for large brood sizes, we ought to see lots of multiple paternity. Slide, please. Okay. So the key assumption, let me remind you, is that there's an equal chance at siring an offspring for each of the mating males. And of course, this is in trying to come up with a null model. Real data don't have to reflect this. So the second principle here is a really neat insight that Ashabibi got. He said, well, you know, actually, we can estimate the chance of siring offspring, and we don't really have to know how many males there were because we do know how many sires there are. And this is because we actually do know the number of tries, that is, you know, for any given species, we'll have some idea of what their average brood size is. And we do know how many successful males there are. And these variables follow distributions. So we actually can back figure what the chance of success would be for a mating male. And if we know the chance of success, then we can construct our null expectation without knowing how many actual mates females have. Next slide. And Hannah, you're on.
So we know that the number of sires in a litter that's of size K is anywhere between 1 and K. That is, we can have up to as many sires as there are offspring in the litter. We can basically describe the distribution of the number of sires using a zero truncated binomial distribution. And this captures the relationship between the litter size, which we call K, that's our number of tries, the number of sires, S, and a probability of a mate becoming a sire, which we'll call Q. Here, this is the equation for the probability mass function of the zero truncated binomial distribution. So this is just describing the distribution of sires. So from data, we actually have K and we have S, but we don't know Q. Also, with this formula, there's no closed form solution for Q, so we have to approximate it. We use Bayesian MCMC simulations to do this. But inherently, by using this equation, or by using that zero truncated binomial distribution, we're assuming that all potential sires have the same chance of success, or the same Q. So in using this distribution to describe the number of sires, we're imposing this assumption that they're mating at random. Mates don't have a different chance of becoming sires, regardless of their fitness, where they live, or what kind of animals they are. So what's the point of having a Q that does not represent the mechanisms behind our real data? Remember, our intent is not to model the real data itself, but to instead compare the real data to a reasonable null model. In order to do that, we need to get a picture of what multiple paternity would look like under our null model. So when we have Q estimated under the assumption of equal chance of becoming a sire, so we're assuming essentially that there are no environmental mechanisms or behavioral mechanisms that influence Q, we can calculate a corresponding probability of multiple paternity under that null model. Here we call it P sub B, and we can get that P sub B for each species in our data from the mean brood size that we get from the literature and our mean estimated Q. So we've given you some equations underlining our analysis, but what are we actually doing? This is a flow chart from our recently published meta-analysis comparing multiple paternity in birds and mammals, but the general concepts that are shown in this flow diagram apply to all of our work so far. So on the left-hand side is our observed data and what we have from the observed data and what we don't have from the observed data. Then on the right-hand side is our null model data. In nature, we expect Q to vary considerably from mate to mate. It can be influenced by things like fitness, environment, and even evolutionary history. In our null model, we ask the question of what multiple paternity would look like if mating happened randomly. So we expect Q to not be influenced by these external factors and be stable across all mates. So what we need to do is we need to simulate data that for all practical purposes are the same as what we see in nature, except for the chance of siring success being equal for all potential sires. If we could access the raw data from the literature, we would actually have the multiple paternity status for every single litter. So which litters had multiple paternity and which did not. And we could actually compare them directly to a simulated version of the same litter under the null model assumption. But what we normally get is summarized data with the number of litters, usually the mean litter size or brood size, the mean number of sires per litter, and the overall percent of multiple paternity across all the litters examined in a single study. We call this sort of the population level multiple paternity because each population is representative of a particular study. We then use the summary statistics of the real data to generate simulated litters that look like real litters. We can then estimate our Q being equal across all mates and calculate a P sub B for the null model for each species. So we can then compare our multiple paternity from the real data to a multiple paternity under the null model. When we do that, we're comparing our P sub B values to the P values from the real data. If they're similar, then the population's percent of multiple paternity is similar to what we would expect had the population been exhibiting mating at random or near mating at random. So essentially no influences of environment or behavior on the chance of success to become a mate. If they're very different, then the population's multiple paternity is quite far from what we expected under random mating. Since K, which is our litter size, and S, our number of sires, are approximately the same between the real and the simulated data, the difference must come from Q. So we need a biological explanation for what changes siring success Q from mate to mate. So remember, we're not trying to actually model the real data. Instead, we're trying to compare the real data to a suitable null model that describes what might happen if each potential sire had an equal chance of success, and then what multiple paternity would look like if that were so. We can then ask which species violate the null model assumption of equal chance of success, and by how much. We can also ask why that's the case, but more on that later. So here's some results. If we estimate what we would expect multiple paternity to look like under random mating for a whole range of taxa, we included fish, reptiles and amphibians, invertebrates, and mammals, this is what we get. So the black line shows the pattern of multiple paternity calculated under our null model assumption across a range of litter or clutch sizes. The x-axis is log litter or clutch size. And then the colored diamonds are our real data estimates of multiple paternity from the literature. So under the null model, multiple paternity increases with increasing litter size up to a threshold and then flattens out. A little like the sort of pattern we see with the number of mates and multiple paternity that Steve showed earlier. In our real data, we see a variety of multiple paternity values at the low end of brood sizes. And we also see more multiple paternity values at the extreme values, either no multiple paternity, 0%, or 100% multiple paternity at large brood sizes. As I mentioned a little earlier, so I showed the flowchart for looking at birds. This particular analysis did not include birds because we were actually reconsidering work by Avis and Liu, which only considered these taxa and did not include birds. So we can actually quantify the differences between the real data and the null model using deviations of P from P sub B. And in our figure, they're represented by the colored streaks. So we have a pretty decent sample of mammals compared to the other taxa in this data set. And many of the populations of mammals have multiple paternity that were actually lower than expected under the null model. So if we look at the average deviations for the null model for each taxa, mammals and reptiles have the largest deviations from the null model. But in this meta-analysis, we had very few samples of reptiles compared to mammals. Also, mammals are viviparous, but the other three groups have a wider variety of reproductive modes, so they're a little harder to evaluate. So in the next few slides, Steve is going to go through some particular mammals that were in these samples and see if we can come up with some biological explanations for the differences that we see between multiple paternity in the real data and the multiple paternity we expect from the null model by looking at some of their behavioral characteristics.
Okay, so we can start with a couple of species of squirrels. And I want to say, first off, we have P, the probability of multiple paternity, and then a G value, and that G value is an effect size. And in the case of the asocial and territorial red squirrel of North America, this G value is negative. And that comes in part because our null model actually is very conservative. So this value actually isn't strongly different from 0. And this is a species where, in fact, we have lots of multiple paternity. There are mating chases when females come into estrus. And little sexual size dimorphism. I'll just mention that female red squirrels are basically running away from the males the day that they're in estrus. And they can mate with several of the males and generally do. Colombian ground squirrels, a species I've spent a lot of time in the field with, are sort of on the edge of sociality. The males are very territorial during the mating season and compete over the females, but populations of Colombians also have fairly high rates of multiple paternity. Here, the effect size, the G statistic, is positive, but relatively small. So by comparison, these have a bit less multiple paternity than we would predict in our null model. So G, when it's positive, is going to show a real difference between the null model and the actual value of multiple paternity in the population. So Colombians, the males are vying with each other for territories, but on the day when females are in estrus, the males actually guard those females, run around and try to isolate them from other males. But the females get away, often mate with other males. Now, in this species, the males are about 17% heavier than the females are. If you look at linear measurements like skeletal measurements, the males are about 4 or 5% larger than the females. So we do see some sexual dimorphism. And I'll come back and make more of a point about sexual dimorphism later. Slide, please. But for now, let's just take a quick look at wild boar, a European species that has fairly large litter size for a mammal and fairly low frequency of multiple paternity. And the effect size difference here between our null model and the actual rate of multiple paternity is really strong. Here we have wild boar living in groups, but when females are in estrus, males guard them. They're not completely successful, but they're pretty successful. And you can see that they're highly sexually dimorphic. The males have prominent tusks. They have erectile mane down the back, and the males are much larger than the females. Slide, please. That's true for brown bears as well. They have a very low litter size and also very low rates of multiple paternity. The effect size statistic is still very strong here. In other words, bears deviate strongly from the null model. They show far less multiple paternity than we would expect, in spite of their small litter sizes. And of course, males guard the females when they're in estrus. Bears are well known for showing yarding behavior, during which the males seldom or don't sleep and try to corral the female. And again, we've got strong sexual dimorphism, both in body mass and in fur characteristics. Slide, please. We can go outside of mammals and see if we can see some similar sorts of differences. These are a couple of skinks from Australia. The southern water skink, actually both sexes are territorial, but there are a lot of individuals that float and don't have territories. Females have lots of mates. Males mate with lots of females. They have relatively high rate of sexual dimorphism, and they hardly vary from the null model. In these skinks, the two sexes look about the same. Cunningham's skink has a different social organization. There are groups of individuals, but in fact, this species is nearly monogamous, and pairs that survive for more than 1 year to breed generally stay together. These have extremely low rates of multiple paternity, and you can see the effect size statistics for deviation from the model is super strong. Males show more colors and spots and have broader heads than females. Slide, please. Here we come to pipefish, and of course, in pipefish, it's the males that carry the young around. They have a brood pouch, and the females can compete to put eggs into the brood pouch. So here we've got a reverse sexual competition situation. So these are actually females that we're looking at. The top one is the dusky pipefish. It has a very high rate of multiple paternity and an effect size that shows that the rate of multiple paternity is similar to what we would predict from our null model. There's widespread mating. Both males and females are drab, so there's little or no indication of sexual dimorphism. Gulf pipefish, on the other hand, have higher rates of multiple paternity. This would be, I guess, multiple maternity. A strong deviation from our null model. The males are relatively choosy. The females compete to put eggs into the male's brood pouch. And as you can see on the bottom, the females are colorful. And what you can't see here is that often the tail fin is strongly banded. Slide, please. So the traditional concept, at least that I grew up with, is you have the polygynous species and then the monogamous species. And the reality is they're all polygynandrous. Both sexes have multiple mates. And it's hardly surprising that there's lots of multiple paternity when you have both sexes mating with lots of individuals of the opposite sex. So species with fairly high multiple paternity, they actually don't require explanation. It's what you'd expect. Species with low multiple paternity, that is a large difference between our null model and the actual value we see in nature, we need some explanations there. So the next step is to look at the phylogenetic comparative method. So far, I looked at a lot of species, but I haven't taken phylogeny into account. Next slide. Okay. The problem with not taking the phylogenetic tree into account is that species aren't independent. In fact, species come in groups of related entities. If species carry a similar trait, then you can have a group where they all carry a similar trait, and you basically have the trait perhaps inherited from a common ancestor rather than independently derived in each of the species. So this is true whether you're studying morphology, physiology, or behavior. You have to have some way of taking into account the fact that similar species have similar traits. So in our case, we're interested in mating behavior, and we have some data on the different species. We need a phylogenetic tree, and then we can map behaviors onto the tree. And I'm going to show you an example of where actually behavior maps pretty well onto the tree. Next slide, please. Okay, so this actually is a pretty neat little cladogram that Konrad Lorenz put together by looking at behavioral displays of ducks. He was interested in ducks and the little dances that they do. We can see a male merganser here, hooded merganser, doing a little bit of a dance to impress the female. And Lorenz took these different sexual displays that ducks show and actually created a phylogenetic tree, that's what we have on the right, for different species of ducks. Now, it turns out that the phylogenetic tree that we have today is pretty similar to what Lorenz put together. The point here is, if you were trying to look at the influence of, say, the environment or maybe some life history trait on these different behaviors of the ducks when they're doing their sexual displays, you'd be in trouble, because in fact the displays follow the phylogeny really closely. So you'd have to find some way to adjust your behavioral analysis for the phylogenetic tree. Slide, please. And Hannah, we're on to you.
Yep. So essentially, we can have things like phylogenetic inertia from traits that evolve slowly, and those can be due to genetic correlations or consistent adaptation. So if we want to analyze the evolution of multiple paternity, we actually need a phylogenetic tree. And we also need to map traits correlated with multiple paternity, such as brood size, number of sires per brood, and even the null model estimates of Q and PB onto the tree for each species. If traits are phylogenetically correlated, they change together over their evolutionary history. If they're correlated independent of phylogeny, they're ecologically linked in some way. So why can't we just consider the phylogeny-free correlations? Well, the problem is if there's phylogenetic inertia, then the species cannot be considered independent observations, which is a really important statistical assumption when we're doing all this modeling. So it's probably best to actually compare our phylogenetic correlations with our phylogeny-free correlations. Typically, many studies use the phylogenetic comparative method to adjust comparisons for the tree structure. These studies typically consider the differences between species due to evolution to be nuisance information, so they remove it and then examine the remaining variation. But we also actually want to know how labile the trait is. So just like the Lorenz ducks, we might have a trait that maps really well and follows the evolution of the tree, and so our species may not necessarily be independent at all. So our first question is, what is the association of multiple paternity traits to the tree? And that's the more important question to ask and to answer before actually considering whether we actually then need to adjust for the phylogeny in further trait analyses. So we actually did a meta-analysis of multiple paternity in birds, and we used mammals for comparison. This is a molecular tree from the BirdLife website, and the colors that are filled into the actual branches of the tree refer to the mean probability of multiple paternity for different species, and that's from the real data. So from this image, it looks a little like birds have lower probability of multiple paternity. We see more blue-purple colors generally in the avian species compared to the mammalian species. So from sort of the mammal sample that you saw earlier, we actually added more species to that and ultimately obtained about 400 studies each for mammals and for birds. Birds generally exhibit much lower multiple paternity than expected under the null model assumption. And remember, the null model assumption is under random mating. And also, that multiple paternity in birds is way less than mammals. You can see here we have birds with a mean multiple paternity of about 0.2, whereas for mammals, we have nearly half of the populations exhibiting multiple paternity. So remember, we want to first know what the association is between phylogeny and multiple paternity, or how closely multiple paternity actually follows the tree. Both birds and mammals seem to show a low to middling association because in this case, like many other correlations, row ranges from 0 to 1. But we actually need some measuring stick to figure out how this stacks up against other traits. So is this actually very low for a typical trait, or is this within normal? So we actually compared the phylogenetic correlation of multiple paternity to that of body mass. This is a trait that's known to be strongly associated with phylogenetic histories. So as an effect size, we found that the association of multiple paternity with the tree is relatively weak compared to body mass. So you can see the body mass phylogenetic correlation is around 0.8 to 0.9 for birds and mammals, whereas our phylogenetic correlation for multiple paternity was around 0.27, 0.28. So this value for multiple paternity is actually within the critical interval of what's expected from randomly generated multiple paternity data. Now, in some cases, we actually have several populations of the same mammalian species. So we also wanted to ask how stable multiple paternity is across populations within a species, which is also known as repeatability. If multiple paternity is highly repeatable, so most populations of the same species have similar multiple paternity values, then the trait doesn't vary a lot from population to population, for example, due to environment. Repeatability in mammals, when we analyzed this, was significant, but not particularly high. So it's about 0.61. So that means about 40% of the variation is not explained by species identity. So that's the variation from population to population. So it seems like we probably should not ignore within species variation when statistically modeling multiple paternity, because we'd actually be obscuring a significant amount of variation in multiple paternity that could be due to population level factors such as environment or adaptation. So this is the beginning of some work with Lauren Hayes and Maddie Roberts. Steve is now going to consider a couple of hypotheses that try to explain the remaining variation in multiple paternity that's not due to evolutionary history.
Okay, I sort of did a teaser a bit with sexual selection before showing you mammals. The idea being that if there's competition among males, that might limit male access to females. So if you have a really dominant male, he's keeping other males from mating with the female. And I'm going to assume for the moment, as some people have working with mammals, that when this sexual selection is on the increase, sexual dimorphism also increases. So in order to look at sexual selection and specifically male-male competition, I make the assumption, and then I'm going to suggest that the more competition you have, the more that would limit the number of mates for females, and you would get lower rates of multiple paternity. And Lauren and Maddie were really good about digging through and finding 56 species, 91 populations of mammals where we could take a look at this conjecture. So we're going to look at the odds of multiple paternity and use the usual 0.05 level, although we're actually much more interested in effect sizes for evaluating our data. So the prediction is sexual size dimorphism goes up, multiple paternity goes down. Slide, please. So here's the model. And I want to take a second with the model because we're trying to get as precise a model as possible. So we're looking at the odds of multiple paternity. We're going to take a logit to do that. We have male body mass over female body mass. That's one variable. That's the one we want to look at. We're going to include here also litter size, because remember, as litter size increases, multiple paternity increases. A here represents our phylogenetic tree. And we also add in the variation among the populations within the species that have multiple populations as another random variable. So both the phylogenetic tree and the populations are taken into account as random variables. And we're controlling for litter size as a fixed factor and looking at sexual size dimorphism. So the result is, for an increase in 1 unit of log sexual size dimorphism, the odds of multiple paternity do go down. Okay, the value that we get, and I'm transforming back from the logit, is about 0.34, and even odds would be at 1.0, so I'm going to compare to 1.0. So as sexual size dimorphism goes up, the odds of multiple paternity does go down, but you can see from the credible interval that it completely covers 1, and this is because there's a huge amount of variation. So in fact, we have insufficient evidence to support the idea that we had about sexual size dimorphism, and I'm serious that we thought that was a good idea before we did the analysis, because we've actually suggested this in our articles. Slide, please. So we need to go on and try some other ideas. And a friend of mine, Jim Brown, got the idea, quite some years ago, maybe 15 years ago now, that a lot of population traits could be explained by the lifestyle of the organism. So this got us thinking about lifestyle. So we're trying to think about how lifestyle might affect multiple paternity. And one obvious way is looking at species that operate primarily in 2 dimensions. These would be things that operate over the surface of the ground, say, versus species that operate in 3 dimensions, things that either use trees, like that tree squirrel that I showed you, or bats, which obviously have 3 dimensions to operate in. So that's one thing we could look at, and I would expect, in general, as you go from 2 dimensions to 3 dimensions, there'd be more opportunities for multiple paternity. Now, another characteristic of lifestyle is the trophic level that species occur in. So we have primary consumers like herbivores, and then secondary consumers like carnivores. And I've done this in a very rough way. I tried to, looking at primary consumers, I actually included a few species that primarily eat insects, thinking that insect food's often super abundant in nature. So as you go from, say, carnivores, which have rare food supply, to an herbivore that has a more abundant food supply, we're going down in trophic level. You might expect density to increase, and as density increases, you might expect there to be more males available to females for mating. So that gives us some ideas of some predictions that we might make about lifestyle. And this is all pretty preliminary. Next slide, please. Okay, so again, we're going to look at the odds of multiple paternity, and I've included in our model here the dimension, whether they operate primarily in 2 or 3 dimensions, the different species, and what trophic level they're at. Now, when I was looking at sexual size dimorphism, I used a continuous variable, and here I'm looking at variables that are categorical. In other words, I'm treating species that operate in 2 dimensions as a 0, species at 3 dimensions as a 1, and similarly for the trophic levels. And again, I've got A here representing my phylogenetic tree and a random term for species that have several populations that have been monitored. And we're comparing to an odds ratio of 1. So going from 2 dimensions to 3 dimensions, the odds of observing multiple paternity goes up 6-fold, and the credible interval is well above 1. So that's a pretty positive result. Going from herbivores to carnivores, the odds actually increased by 1.25. Now, I predicted that the carnivores would have lower odds of multiple paternity, not higher. So this actually is a result in the wrong direction, but it broadly overlaps 1.0, so it's not significant. So our preliminary conclusion is that there's some evidence that the number of dimensions that species use in their lifestyle, but also with respect to mating, is supported as an idea about why we see differences in multiple paternity. And in fact, we don't have sufficient evidence for a difference due to trophic level. Slide, please. Okay, so in conclusion, I'll wrap up for both of us. Although Hannah, you can jump in anytime you like. Multiple paternity is pretty common among species where there's more than one offspring at a time. But we need a null model to figure out, well, just how much multiple paternity should we see in nature? I think when we first saw multiple paternity, we were pretty surprised to see it at all. But in fact, I think we ought to see it quite broadly in nature, and in fact we do. So we can make a prediction based on the litter or brood size and the mean number of sires in different populations. We can get an observed value, develop a null model, and look at the difference between our observed value and our null model, and get an idea of which species are going to be most informative in terms of trying to understand why multiple paternity is occurring in populations and what causes variation in multiple paternity. And in a sense, we've turned our questions about multiple paternity on its head. And I think this was particularly true with people working on birds who started noticing multiple paternity and said, oh my God, how can that even occur? Well, our question, having gone through this exercise, is how come we don't see it more often? Why don't birds have a lot more multiple paternity than they do? It looks like they don't have much compared to either our null model or to the mammal data. So that's an important thing to explain and a good topic for future research. Multiple paternity is only weakly associated with phylogeny. And there's some question about when one should or shouldn't take phylogeny into account. You know, a lot of people will say, well, geez, if you can take it into account, you should just do that and see what you get out of it. So for people who feel that way, I guess that's great. But another friend of mine, Trevor Price, 25 years ago said, you know, you really should run these analyses with and without the tree. You know, that might tell you something. So let me just say, we've only shown you things where we took phylogeny into account. We ran these same analyses without the influence of phylogeny taken into account and got the same results and would make the same conclusions. I do think, though, that variation among populations really matters, and explaining some of that variation could be really informative. It looks like multiple paternity and the odds of multiple paternity are pretty flexible from population to population. We did do a preliminary test of our first idea about male-male competition, and that actually looked pretty unpromising from an analysis of mammals. And in future work, we think it's important to examine more ideas like the lifestyle traits that we looked at. And if anybody has additional ideas, we'd love to hear about them. Slide, please. Now, I need to point out that much of the work that Hannah and I have done has been done in Africa. And this is at a conference that occurs annually that's organized by Overton Genda. This is called Masamu conference. It's basically a study institute for looking at application of math and statistics to biological problems, and potentially other sorts of problems too. And it's sponsored by NSF, so we have to thank them for sending us to Africa to think about multiple paternity. And I've just put a few extra species here that have high rates of multiple paternity among the mammals that we know best, but we'd be happy to answer questions.
Thank you, Steve and Hannah. That was really quite fascinating talk. So at this time, it's time for questions. And just a reminder, if anyone's new here, we'd really like to have questions, and we encourage particularly students and postdocs. If you're a student or anyone has a question, please type a question mark in the chat. If you're online on YouTube, please type your question or comment in the chat function there, and one of us will forward that to the speakers. And again, if you're a student or postdoc and you have a question, you can put an S or a P next to it so that we can push yours up to the top or closer to the top. And also, reminder that as we try and build community, please tell us who you are, what your interests are, and where your institute, where your affiliation is. So with that, I'm going to start questions, and we'll start with Maren, please.
Hello, and thanks both for your very nice talk. I have one comment and one question. The comment is about your assumption that with increasing sexual competition, there should be more sexual size dimorphism. This is something that I do not agree with, because I think the question is the sexual competition only in one of the sexes, or is it in both? Because you can have very strong competition in both sexes, and then there is not necessarily a reason why you would expect sexual size dimorphism in the two. And do you want me to pose my second bit later after somebody else has talked?
Well, let's have the first question answered, and then please have a follow-up. We've got plenty of time.
Okay, Hannah, why don't I take this?
Yep.
We didn't test your hypothesis. We looked at what we felt like was a reasonable idea given the literature, but we weren't asking your specific question. I will say that if you turned it around and asked the question the other way, sexual size dimorphism, as near as we can tell, doesn't have much to do with multiple paternity. So it kind of doesn't matter which way you answer the question. And we could be very wrong about the assumption that we made. It's an assumption. We're not claiming it's true. It's an assumption. Okay, please go on.
Okay, my question is whether you actually took the window of fertilization, so how long an egg cell can be fertilized, because if there's multiple paternity, but the window how long the egg can be fertilized is very narrow, then it doesn't matter whether the female is mating with 50 males if she mates only with one during the 1 hour where the eggs can be fertilized, for example.
Hannah, you want to take that, or you want me to?
You can go ahead, but the essential answer is no, we didn't account for that.
We didn't account for it. We have thought about that, but I want to put that in the context. That's an explanation for why you may get low multiple paternity. So that's an additional hypothesis. We have thought about that, and we've thought a lot about precedence effects, which
Yep.
would be associated with that. But we've not tested that yet.
Okay, thank you. Next question is Eugenia.
Hello, thank you for the very interesting talk. I work on domestic dogs and domestic cats, sexual strategies, but I underline that it's both of them free-ranging, so they are free in their movement and in their mating. And we did find multiple paternity in both cats and dogs. And one thing that you did not mention is that it's very important, the synchronization of females. I mean that, of course, how big the dimorphism can be. A big male cannot control 10 females if they are synchronized in their estrus. I mean, both territorial male or alpha male. So I think that it can be a very important life trait for the result of the rate of multiple paternity. Oh, it's more a comment than.
Steve, go ahead.
You're probably familiar with Dominique Pontier's work on cats.
Yeah.
Yeah. You know, that Dominique's work is one of the earliest things really nice works that I saw in multiple paternity. It's one of the things that got me interested in the topic of multiple paternity, other than my squirrels have multiple paternity. So I think that's also a great hypothesis to look at. We'll have to figure out how to differentiate predictions from sexual competition among females with things that would restrict the number of males that can access females. Sexual selection is a big problem because, you know, females may only choose to mate with one male. And then you don't get any multiple paternity. So it'd be nice if there was some way of teasing out, you know, the attempts at males to sequester females, and then female choice. I think female choice is going to be the harder one to deal with. But sexual selection is especially difficult because you have these two aspects that you can think about.
Thank you.
All right, so we have a student question that just popped in, so I'm going to jump ahead, Eduardo. Is Evie or Evie? I'm sorry if I said that wrong.
Or Eve. Hi, everyone. Thanks. I'm a postdoc and researcher in France at the ISEM, the Institute for the Evolutionary Sciences here in Montpellier. Thanks for this very interesting talk. I kind of have a comment reacting to the coercive potential of males and the possibility for females to express maybe mate choice. I thought it was quite, I also think first that indeed there might be something to look at in the coercive potential of male, whether it's because they are larger than the females or because of the ability of the females to express counter strategies, and this possibly will influence the ability of males to monopolize that, of one or a few males to monopolize access and paternities with the females. And I think maybe it is partly what you see when you looked at how did you call it, the 2-dimension or 3-dimension of the landscape of the animals. Because it seems, at least as far as I understand, that animals navigating in 3D dimensions, whether it's in the ocean or in trees, for example, usually males have more trouble to sexually coerce females. So it could be a reason why there is more multiple paternity in these environments. Then I was just curious to know if, I don't know if it would fit in this framework or not, but if you considered also multiple paternities through time, like when females mate over time, have a long lifespan and can have different mates with different clutch or litters?
Yeah, so I'm going to speak as best as possible to the behavioral aspects. My background is more in statistics and modeling, and Steve's dropped off for a second. I think his signal's poor, but these are all great ideas, and certainly these are some of the theories we've been thinking about to explain multiple paternity. I will point out that actually, I'm sure many of you are looking at these analyses and thinking, well, why didn't you consider these as parts of the questions in the first place? And I think just to give a bit of context, I think Steve read a few of Avis and Liu's papers on multiple paternity in animals. And one of the initial hypotheses that Avis and Liu had put forth is that the relationship would be linear, and that essentially for increasing size of the litter or brood, we would have increase in multiple paternity. And there was no cap to this. It would just continually increase. And so essentially that's what prompted us to think about, well, are we actually modeling multiple paternity correctly first? And what if the relationship is not actually linear? We found it was not. It actually seems to have a curvilinear relationship and have this cutoff point. And then secondly, if it's actually curvilinear, we're thinking about when people examine this question of multiple paternity, like Steve said, they're usually thinking about, well, you know, we shouldn't see it, so when we see it, it's surprising. And what we realize is that very often, so in statistical models, when they're translated to ecological hypotheses, it tends to be a comparison of no effect. So I think this was brought up in the last seminar, actually. A lot of the biological hypotheses tend to be looking at no effect. And we thought, actually, this is not the kind of question you want to ask with multiple paternity. We actually want to think, okay, so in behavioral studies, we're thinking about what influences multiple paternity and what would it look like if we actually removed all of those influences. And what happens when you remove those influences is not no effect. You actually get no effect of behavior on those influences, but you don't necessarily get no multiple paternity. You actually get lots of multiple paternity. And so that's where the modeling structure came in the first place. And it's only been more recently with our work with Lauren, for example, that we've actually been exploring these ideas of why we actually might see less multiple paternity than we should and what those mechanisms are. And then also trying to quantify that in a way that we can actually put in a model, which is the more difficult aspect of it. So Eve, those are excellent points, and those are things that, you know, we'll probably be considering as well in our subsequent analyses.
Okay, so next we have Eduardo.
Thank you, Eduardo Fernandez Duque, Yale University in the US. Thank you, Steve and Hannah. I really enjoyed your talk. And first, I want to thank you. The first few minutes, you walked us slowly through some topics that maybe most of us, don't think about it regularly. And one of them was really working so hard on pointing at the fact that you're going to make assumptions explicit. So I understand Magen's comment, but I think that part of, and now Hannah, you were making references to the talk last week. I think the problem we have in our field is that we think, not because nobody discusses the assumptions, we're not making them all the time. So suddenly, we're right. We need to get into, these are all the assumptions. I'm going to make them explicit. Maybe I can even test with sensitivity analysis how much my results change. So let's not forget that every single time we read about any test with some associated parameters, there are several assumptions that the researchers are not even pondering. So thanks for bringing this forward, and we should be discussing the assumptions and seeing what we learn from making them and then modifying them accordingly. So that's a comment. The question is, some of us, many in the audience, have been working hard in trying to get at primary data for analysis that cut across taxa. It's great that now you're going to be working with Lauren and Maddie on a data set that they've been curated so carefully. I was wondering, can you share a little bit, how did you go about deciding on the data to use from the meta-analysis? I think it was Stephen who explained that, yes, what we bring from the field and we produce an estimate of the probability of a multiple paternity. But my suspicion is that most studies that you're going to find out there, after this continuous variable, that is the probability, make a decision, a dichotomous decision. I might call it multiple paternity or not, and that's what makes it into the published record. And in our own research, we're now struggling to decide what to do with estimates of paternity for the probability in a pair living monogamous primate of having more than one male siren offspring. So did you go through the specifics of how in each study, because if some people are deciding to go with multiple paternity with a P of 0.8 and some others with a P of 0.9, because that really influences whether you get published or not, then that is going to bias completely your analysis.
Yeah, so we actually had some problem with that in the birds samples that we use particularly, because figuring out multiple paternity in birds can be quite complicated. And it also depends somewhat on the molecular techniques used.
Right.
So we actually, many of the mammalian studies tend to use the DNA microsatellite. But in birds, we actually had quite a few that used fingerprint studies. So in our meta-analysis of birds versus mammals, we actually looked at whether there was a significant difference between these two types of procedures for estimating paternity. And essentially, we found that there was a difference. Fingerprinting detects it a lot less. So since the microsatellite analyses are more recent, they're more technically detailed. We use studies that were only from microsatellite analyses, and that's where most of the analyses of the sort of birds versus mammals comparisons came from. So that is something that we thought about. However, for most of the studies that we're looking at, we don't actually, especially some of the older ones, we don't actually get the raw data. So we don't know what their cutoff point is for determining whether there was multiple paternity or not. We just get at the very end, here's how many how many broods we analyzed, how many of them had multiple sires, and how many sires, so they'll usually say, okay, well, 10 litters had 3 sires, five litters had 7 sires, or something like that. That is pretty much all we get in many of these analyses, and we have to then calculate multiple paternity for those just from those summary statistics. So in most cases, we didn't actually go back and look at the individual cutoff points for when they're actually detecting multiple paternity and what their cutoff points were. But that's something to consider, because again, like we saw with fingerprinting and microsatellite, there is actually a difference in detecting that important value, multiple paternity, in the first place.
That's very interesting. I imagine that once you tackle the same questions with data sets that are going to be much better curated, because these are different times, like Lawrence and Maddie's. If suddenly you find that when I look at data for which I have all the nitty-gritty details, estimates go up and down, that may be informative to some extent, or at least raising doubts, particularly if you're looking at the similar taxa, right? Thank you so much, Steve and Hannah. And I should say, when Steve asked us, can we do a combined 2-person talk? I think I was the one who I wasn't thrilled, but you have shown us the way. I think it worked out great. So thank you for doing it.
Oh, great. Thanks.
All right, so I have a question from a student on YouTube. So I'm going to go with that first. Then I'll go use the layman, okay? So this comes from Miguel Angel Torres. They're from Rosario University in Colombia. And they were wondering if you think multiple paternity varies based on the different, whether they vary between the temperate zone and the tropics due to differences in seasonality.
So that's something that we were actually talking together before the seminar. So one of the things that we see in multiple paternity is this reasonably high variation within certain species. And for especially birds, for example, their ranges can vary quite significantly across the latitudes. And one thing that we could potentially consider is, temperature variations. Steve, for example, works on Colombian ground squirrels. And one of the things that he's been studying over the 30 years of looking at them in the field is how their emergence changes with climate change. So as we get earlier springs, they emerge earlier. And so timings of matings would change, and possibly the lengths of those timings would change as well. So this is something we've been thinking about in terms of looking at maybe whether there's differences in latitude gradients or other environmental factors that might actually explain these differences within species.
Great, thank you. And Miguel, if you have a follow-up, please post your next question in YouTube and I'll forward that on. So next we have Zulema. Zulema, are you ready?
Yes.
Excellent.
Just barely, but yes. First of all, Zulema Pang Martinez retired from the University of Missouri St. Louis, professor emerita. I really liked the talk. I thought it was incredibly thought-provoking. And the one thought that I had about the issue of mammals having higher levels of multiple paternity versus birds, it reminded me of a paper that Patty Gawadi wrote many years ago in a different context, looking at forced copulations. And she talked about the fact that female birds, except for the species that have intermittent, where males have intermittent sexual organs, the females are pretty much able to control whether mating occurs or not, because of course they have the cloacal kiss, and females can simply refuse to expose their cloaca by keeping the tail down. And so I couldn't help but wonder if there might be, if that may have something to do with the fact that birds tend to have fewer cases of multiple paternity, if females are actually using that option to keep additional males from mating with them. And I have to say that also, like Maren, I think in all of this, it's super, super important to keep in mind the female contribution to all of this phenomena. And obviously, we're now beginning to realize that there's a lot more female-female competition that we had sort of overlooked because it's so different from male-male competition in sexual selection. And I think that trying to always keep that female perspective is going to be really critical to being able to understand the phenomena we're talking about.
Absolutely, I agree. And I think, I mean, your point about birds is definitely interesting, and I think complicated to answer in a way because of the fact that essentially, if we look at the division between birds and mammals, we also have a huge evolutionary diversion, right? So that becomes complicated in the sense that a lot of the division between the two can be explained by the evolutionary history, and those divisions are essentially obvious. And I think if we could find a way to answer that question, say within birds, we might be able to find more characteristics that indicate that, you know, female choice has some influence on the ability to have multiple paternity in populations. But it's a very complicated question to answer, but it's definitely something that we've thought about in terms of those differences between birds and mammals, beyond just, you know, they're huge differences in mating behaviors and things. So
yeah, and with regards to, I think I was just coming back, and so I'm not sure I caught it all, but I think that I think there was a question about the tropics and birds in the tropics.
Yeah.
Again, years ago, Regina Macedo, who's from Brazil, sent me a paper, which God only knows where it is now, that that actually, I think, showed that there was a difference in terms of extra pair copulations and I think more extra pair fertilizations between the birds that breed in temperate regions versus the tropics. And I know at the time we were talking about brood size and whether there's a confound because the larger the brood size, statistically, the more likely investigators are to find evidence of multiple paternity, those sorts of things. But yeah, I think there is a difference, and I want to say that there's more multiple paternity in the tropics, but it could be the other way around. I just can't remember right now.
Yeah, that'd be definitely interesting to follow up on because that's something that we could actually incorporate into our model now, where we actually control for the brood size, control for their evolutionary histories, and then look explicitly at their different temperature zones and see whether there's actually a difference. So it'd be interesting to sort of reanalyze that question with more data, new data now, and see what we find from that.
Okay. If I find the paper, I'll send it on.
Please do.
Because I have Steve's email.
That'd be fantastic. Yeah. Thank you.
It's somewhere. The pandemic interrupted. I was moving into a new office when the pandemic hit. And so I still have boxes and boxes in my quote unquote new office that I've never gotten around to unpacking. But I'll see if eventually I can find it.
That'd be great.
Yeah, thanks a lot. It was really cool seminar.
Thank you.
Very, very interesting.
All right. Thank you. So next we have Emmy.
Hi, I'm Emmy. I'm from the Max Planck Institute for Biological Intelligence near Munich, and I work on extra-pair paternity in blue tits. I really, really liked your talk and the paper, and I really like your approach of turning this question on the head and asking why don't we have more multiple paternity instead of saying why do we observe multiple paternity. I think both questions have their merit, but one has been asked a lot, so I really, really like this novel approach, and thinking about a null model is also something I really appreciate and I think is super important. So thanks for that. I have one comment, or actually it's two comments. One is about when you're working with those more, I think Eduardo called it more curated data sets, possibly you also have not only information on multiple paternity per se, but on the precise number of sires. And I think it would then get much more exciting even than it is already if you don't have this dichotomous trait, multiple paternity or not, because obviously it's a continuum and you're sort of artificially imposing this because the data report what they report. But if you have data which do provide that information, it's probably nice to use that and then extend it to n number of sires instead of multiple paternity. I think that would be really cool, and then see how in these more curated data sets ecological variables or something would explain that. And then about these variables which could explain deviations, I was just wondering, I have a comment on this carnivorous herbivorous contrast, because when it was presented, my intuition was that multiple paternity would be more common in carnivorous species. And my reasoning behind that, I guess, I mean, it's more of a gut feeling, but I guess my feeling is that for a herbivorous species, foraging opportunities are spread out, so things are less clumped, and therefore social interactions might be reduced to the extent that you're lucky even encountering one opposite sex individual at all. I'm thinking about shrimp or something like that. And then for the carnivorous species, you would think maybe food sources are more clumped and maybe they even hunt in groups or something like that. And then social interactions are more common, and you would maybe expect an increase in multiple paternity. That's just a comment on that contrast, because it was presented the other way around, which was also a plausible argument. I guess you can read it both ways around. I just wanted to add that. So thanks again.
Sure,
Very
thank you,
exciting.
Emmy. So related to the second comment, I think that so in this particular case, we were only looking at mammals, and I think it's possible that, for example, our hypotheses might change depending on which taxa it's in. For example, fish have very different environmental spaces that they occupy, and very different behaviors associated with that environmental occupation. So the hypothesis can certainly change. I think back to Eduardo's point, it's important to basically just say what our assumptions are in the first place. And if people disagree with our assumptions, then that's even better. That's something that we can actually have a dialogue about scientifically and consider other options. In terms of the herbivore and carnivore, even if we had the opposite hypothesis for the data that we looked at, the effect was not significant. So essentially, we see no effect. But like I said, this could change based off of who's looking at the question, and also the taxa that you're analyzing. And remind me briefly of the first point again. I had a comment with regards to that.
I just mentioned that extending this beyond a yes/no multiple paternity number of sires would be super cool. That's right. Yes. So I don't think this was posted on the must reads, but we actually have another paper with Abibi et al. With our collaborator Asha Bibi, where we explore through simulations this relationship of sires, mates, which is something we can technically obtain in terms of an approximation if we assume that Q is equal among all sires. So this would be under the null model. But we actually explore these sort of trade-offs between number of sires, number of mates, litter size, at least theoretically under the null model. And it'd be super exciting to actually take those theoretical results and actually look at real data and see how those play out as well. So I absolutely agree with you on that point. Thanks again. Thank you.
All right, so next we have Anna Marie.
Yeah, hi. Thanks for, great talk. My name is Anna Marie. I'm at a postdoc at the Universidad Católica in Chile, but I've worked with Steve in the field with the Colombian ground squirrel. So I remember watching the mating patterns and everything. So that was fun. So Steve also kind of like explained in his part of the talk where he was kind of like talking about which kind of mating or like reproductive strategies they had, mate guarding, or they were territorial, mate chasing. Do you see kind of like a pattern of like the amount of multiple paternity and what kind of like mating strategies there are?
I don't know whether Steve has looked at that. I know he's played around with these kinds of thoughts more analytically. So I have not seen any results from that if he's played around with it a little bit to explore it. But that's something that we have talked about in terms of potentially there being a difference. I think that's where his original idea about sort of the sexual size dimorphism came about, was summarizing that into a more dichotomous grouping where we could just basically lump these into more larger groups and then ask the question. But we haven't actually looked at categorizing each of the species explicitly. And I think some of that is also we have a limit on sample size. So in most of the analyses that we've done up to this point, we're talking about anywhere between 50-ish to 60-ish species of mammals. And the problem with that is if we have a variety of different mating behaviors, we probably only have maybe 2 or 3 mammals per group then. So we lose a lot of sample size then. So it'd be interesting to ask that question, but we might have to summarize them more generally so that we have sufficient sample size to actually answer that.
Thank you.
Thank you.
Okay, so I'm looking both at YouTube and the queue here. I don't see any more questions. Does anyone else here have a question they want to put in? If you have a follow-up? I see that Steve is back, but I see his microphone and camera not working. Well, I don't see any more questions. So what I'm going to do is I'm going to stop the live stream to YouTube. And of course, we can sit around and chat for a while. So I just want to thank both Hannah and Steve for a really wonderful talk. And I agree with everyone that here that, with Eduardo in particular, that the describing the methods that carefully really was helpful to me. So I appreciate that very much. And again, thanks, Hannah, for running with this when Steve got dropped off. So I appreciate that as well. And thanks for being here.
Thank you. Well, thank you for having us, and I appreciate your patience with my less than perfect answers for some of the biological mechanisms, because I tend to do a lot of thinking about the models.
I think you did fantastic, so I appreciate that.
Thank you.
All right, everyone. I hope if people want to stick around, we can stick around, but I'm going to stop the live feed for those of you watching. Hope to see you next week when